Most of us have seen heartwarming stories of family members reunited from afar by DNA matches. Here we look at forensic applications of the groundbreaking technology, with mixed verdicts on its safe use moving into the future. Criminal tracing. A truly beneficial application of the new DNA technology is its use in tracing and capturing dangerous criminals. The so-called Golden State Killer was apprehended in April 2018 after detective work was integrated with DNA databases and family lineages to indicate potential suspects, a method known as genetic genealogy. Joseph D'Angelo, now 74, had used his knowledge as a former police officer to avoid leaving evidence and successfully escape justice for over 40 years. However, he couldn't conceal its clues in his own genes. D'Angelo has since pleaded guilty to the slaying of 13 people, the assault of around 50 women, and staging burglaries across California during the 1970s and 80s. The process of genetic genealogy starts with profiling the perpetrator. Investigators collect biological material like blood, hair, skin, or semen from a crime scene, samples which sometimes contain DNA. This can be read via genetic sequencing, where the DNA is cut into tiny fragments which are scattered over a genotyping chip to see what adheres. The chip holds microscopic wells with about 700,000 probes that each match a distinct genetic variant, a particular sequence that may, with luck, be found in a person's DNA. Fluorescent dyes are then applied in isolating the appropriate probes so that a computer can identify the sequence of DNA letters in a sample, thereby creating a genetic profile. The next stage involves locating relatives of the perpetrator, whose sequence is compared with a public database of DNA sequences, such as is found on genealogy websites. These can be searched to find similar profiles amongst millions of users. Law enforcement officials can use the databases to determine whether this suspect is closely related to other people in the database, as calculated from the number of shared genetic variants. A person's DNA is roughly 50% similar to each of their parents and 25% similar to grandparents. For each generation after two individuals shared a common ancestor, such as grandparents, their genetic similarity is reduced to 1/4, meaning that first cousins share about 12.5% of their DNA, declining to less than 1% for third cousins. The family tree of the perpetrator is thus painstakingly constructed. Unless all of his close relatives are ancestry fanatics, it's highly unlikely that many first or second cousins would be found in the DNA match database, but thousands of third cousins could be found. Investigators in the Golden State case focused on third cousins. The traditional method of genealogy, locating old records like birth and marriage certificates, census information, and newspaper obituaries, used in combination with modern tools like Facebook stalking, are then integrated with the DNA profiles to construct a huge family tree of people who could be related to the perpetrator. The family members are added one by one to the tree in a process like a reverse engineering genealogy. After investigators have identified some potential suspects using these genetic links, they pursue conventional strategies for identifying the suspect. These include procedures such as comparison of current physical features with previous eyewitness statements and police sketches. In this case, the choices were finally narrowed down to two candidates, and D'Angelo's incriminating DNA was clandestinely collected from his car door handle and a tissue found in his rubbish bin. While some concerns have been raised about the ethics of the secondary use of personally identifiable information, there can be no denying the beneficial outcome of the perpetrator's final capture and life imprisonment. Cold cases. Another heartbreaking crime has been solved by DNA analysis, but 75 years too late. In 2017, Audrey Bell, a 51-year-old mother of triplets from Long Island, purchased online a DNA testing kit to enable her to work out which of her triplets were the identical siblings and which child was the fraternal sibling. The results puzzled her. While the test was able to correctly identify who of her triplets was the fraternal sibling, and their heritage was categorised as Southern European, the ancestry composition made no mention of anything specifically about Italy. That struck Belle as strange, because her father, Richard Palmadesso, had always been very proud of his Italian ancestry. Her twin, Cynthia McFadden, also later undertook DNA testing, which again made no mention of Italian ancestry. By then the twins' father had passed away, so they asked his first cousin, also called Richard Palmadesso, to also take a test, which astonishingly revealed that they were not related. Cynthia had requested the ancestry site to indicate any DNA relative connections in their database and found that she had matched with a man called Tom Martin. It was revealed that the twins shared 22% of their DNA with this man, meaning that he was either their grandfather or uncle. They contacted Martin through the website, finding him to be a 79-year-old retiree living in Florida who had registered for the test for a very specific purpose. He had been trying to locate his missing brother Gerald, or Jerry Martin, for decades. Tom related to the sisters that on the July the 9th, 1945, Jerry, just 2 days away from his 4th birthday, was kidnapped when he and then 6-year-old Tom were out riding their bikes together. The incident occurred close to his home in the Hell's Kitchen neighbourhood of New York City. Tom remembered the two boys being approached by a woman offering them candy. Jerry left with the woman to fetch the candy while Tom stayed by the bikes, but Jerry never returned. At that time, there was no rigorous missing children's registry or system of searching. However, a common kidnapper profile then was a woman unable to have children who experienced pressure from a husband or society to produce a child, often to maintain her relationship. Some adoption agencies even turned to kidnapping. With no DNA testing available at the time, no one needed to be any the wiser that the children being raised in a particular family were not biologically the parents' own. The striking similarities between Tom Martin and the sisters' memories and the images of their father, even down to tastes in food, confirm what the DNA had already revealed. They pieced together a possible narrative to explain the swap. The twins' grandfather had served in World War II for around 2 years, arriving home to Staten Island in September 1945. The two women speculated that it was possible that their grandmother had become pregnant just before her husband left for the war, and possibly miscarried. Or, for whatever reason, she may have even lied to him that she was expecting. With the Nazi surrender to the Allies on May 7, 1945, and her husband's return imminent, she may have felt compelled to produce a 2-year-old son, leading to the kidnapping of a boy who seemed to roughly approximate one of that age. Social control. In an admirable not-for-profit campaign started early this century, bearing the name Hong Kong Cleanup, an ad agency created eerily realistic images of outed litterbugs. Even more scary, the techno-futuristic Most Wanted style portraits were created from DNA extracted from actual trash. In Hong Kong's 2015 Face of Litter campaign, the agency creatives teamed with Parabon Nanolabs in Virginia, who had developed a process for constructing digital portraits from tiny traces of DNA. Parabon had begun developing the technology more than 5 years previously in collaboration with the US Department of Defense, mainly to use as a tool in criminal investigations. By analysing a mere nanogram of saliva or blood, the company is able to make a fairly reliable prediction of your facial characteristics. The DNA found on harvested Hong Kong rubbish was sent to a genotyping lab where a huge data set on the litterbug is generated. The data is processed with Parabon's machine learning algorithms and starts to build a rough snapshot of specific phenotypes or traits. The company hones in on what is described as highly heritable traits, or traits that are subject to the least amount of environmental variability. These are features like eye, hair and skin colour, freckling and face shape, which are far easier to determine than height, age and even hair texture. The team also factor in characteristics like age. For example, young people are more likely to chew gum, while cigarette smoking is more common in the 45 plus age group. Given that the ethics of the creepy images are questionable, Parabon states that it gains the permission of every person whose trash they process in this way, and so avoids the accusation of unsolicited public shaming. Information and bioartist Heather Dewey-Hagborg has set out to show how arbitrary these images are. Concerned about the prevalence of surveillance technology, she used a 3D printer to produce the life-size 3D portrait from DNA in discarded items she had found, such as hair, cigarettes, and chewing gum. However, perhaps to highlight the subjective variability of the technology, she constructed an installation of 30 portraits of transgender whistleblower Chelsea Manning. The facial sculptures are all based on cheek swabs and hair clippings that Manning sent her while imprisoned, and show the huge variation in the results that can be obtained. The artist selected 2 portraits that most resemble Manning at the time, but the huge variation in the other images shows how subjective the technology is in creating likenesses.